Welcome to Biology My Passion. I am Soumya Harikrishna. Today we are learning the chapter Photosynthesis in Higher Plants. Photosynthesis is one of the major processes happening on Earth and it is very significant. So from the word itself, photosynthesis. Photo means light. Synthesis means manufacture. So manufacturing in presence of light, that is called a photosynthesis. Who is doing this? Green plants are capable of doing this, or some photosynthetic bacteria also can carry out this process. Now this process is the physio-biochemical process, the only process which fixes atmospheric carbon dioxide in the form of carbohydrate, that is food. Every organism in this world needs nutrition or food for energy. So from where are we getting this? We all depend on plants for the same. Why are we depending on plants? Because plants only have the ability to prepare the food. Prepare the food means what? They only have the ability to capture the sunlight and convert it in the form of chemical energy. So when I am consuming the plant, I am stealing that chemical energy stored in the plant. Then later I utilize it. So if I am using the energy now to talk to you or while teaching you or doing some work, from where did I get? I got it from plant. From where did plant get? Once upon a time the plant captured it from the sunlight, indirectly I am using it. So ultimately if you see, there is only one source of energy in this world, that is our sun. So every organism is deriving energy from the sun. So plants can directly derive it, we cannot take it, so we wait for the plants to take it so we can steal it. Okay, so this is how the photosynthesis is functioning. Now the photosynthesis is very significant. Why? Because it has got two importance in the universe. That is, one is it is the primary source of food on earth. Rest all organisms depend on plants because they do photosynthesis. And second is, photosynthesis is the process by which oxygen is released into the atmosphere. We know that all aerobic organisms keep on breathing oxygen. So imagine if there is no oxygen replenished, what happens? Oxygen will get over, right? But photosynthesis is the process by which plants keep on releasing oxygen so that oxygen is not getting over. So because of these two, photosynthesis is very, very significant. Now, what is happening during this? Organic food is produced. Organic food in the form of carbohydrate or glucose, we can say, the simplest form. Then it will be stored in the form of starch. But from where is it forming? From inorganic substances like carbon dioxide, right? So two raw materials are required for photosynthesis, carbon dioxide and water. So the inorganic materials are converted into the organic food during photosynthesis. And two more conditions are required. One, a pigment to capture the sunlight, that is chlorophyll, and also sunlight, which is the source of energy. We learned that these four things are needed for the plants for photosynthesis, that is carbon dioxide, water, sunlight and chlorophyll. So in class 10, there were many experiments to prove that each factor is necessary for photosynthesis. Suppose if I want to find out photosynthesis, a particular factor is needed for photosynthesis out of these four: water, carbon dioxide, sunlight and chlorophyll. Suppose if I want to check to see whether carbon dioxide is necessary, what should I do? I don't give carbon dioxide and see whether photosynthesis has taken place or not. If photosynthesis is happening, of course carbon dioxide is not necessary. But if photosynthesis is not happening, carbon dioxide is a must. The same way, if I want to check sunlight is necessary, don't give sunlight. If photosynthesis is happening, okay, sunlight is not required. If photosynthesis is not happening, sunlight is a must. This is how we are checking. But how will we find the result whether photosynthesis has taken place or not? We have to see the product. Starch is formed or not. If starch is formed means photosynthesis has happened. If starch is not formed means photosynthesis has not happened. Right? How will you test for starch? Using iodine. But here there is a problem. When I use iodine, stored the form of sugary starch. So it may be the previous one stored. So to get a clear result, we have to first destarch the plant. So before doing any photosynthetic experiment, the first step is to destarch the plant. If you keep the plant in some dark room for 2 days, means 2 to 3 days, all the stored form of starch will be utilized. Once you take up, this plant is de-starched plant, means all the starch has been used up. Now you can start your experiment. So this is the process of de-starching the leaves. Followed by that, we can do our experiment. Suppose I want to do the experiment to prove that sunlight is necessary. What should I do? I cover a part of the leaf with a black paper on both the sides and keep it in the light for a few hours for the photosynthesis to happen. After that, I remove the black paper. We are doing the starch test for the leaf by putting iodine in it. Only the part that was covered may not show the blue color, whereas the rest of the part shows blue color. This proves that photosynthesis requires light. The same way, if you want to find out the importance of chlorophyll in photosynthesis, you have to take a variegated leaf, means a different colored leaf. And you can mark which all parts are green just for your reference later. Then keep it in sunlight and see later the adding iodine solution. You will find that I can match with the leaf trace that you had already taken, that whichever parts you mark the green only will be turning blue green, rest blue black, and rest all will be colorless. That means only the green part could carry out photosynthesis. That means chlorophyll is also necessary for photosynthesis. We also conducted experiments to find that carbon dioxide is necessary for photosynthesis. How did we do that? That is, we have to take again a potted plant, discharge it. After that, we are keeping half of the leaf inside a KOH solution. KOH pellets or solution, potassium hydroxide, it can absorb all carbon dioxide. So the part which is inside the test tube containing KOH does not get carbon dioxide. Later when you do the starch test, that part may not show blue-black coloration. From that we understand carbon dioxide is also necessary for photosynthesis. Now we will discuss certain early experiments that led to all these knowledge that we have about photosynthesis. It was not one fine morning somebody found out all these factors, all these processes happening during such a complicated process. But it was the work of several scientists over a period of 200 to 300 years. We got the overall idea about it. But the work started as early as 18th century, that is 1770. A scientist called Joseph Priestley, he discovered something about a photosynthesis. Joseph Priestley is actually known for discovering oxygen. He discovered oxygen after 4 years, that is in 1774. But when he was doing this experiment, he did not know about oxygen. So what was his experiment? He took a bell jar. Inside that he kept a living organism, that is a mouse, and a candle which is burning. We know that now we know that for a living organisms to live, oxygen is essential. For a candle to burn also, oxygen is essential. Then he closed that bell jar and sealed it airtight. So no more oxygen could enter. But after some time when he observed, he found that the candlelight extinguished. At the same time the mouse also suffocated inside. Then he did the experiment by keeping a mint plant also inside the bell jar. This time he found that the mouse was not getting suffocated, and also the candle continued to burn. From this he summarized that the burning candle and the living organism mouse fouled the air, but that was restored by whom? Green plants. Okay, but he did not know that the green plants were giving oxygen, though he discovered oxygen later. This experiment was later continued by another scientist called Jan Ingenhousz. What he did the same experiment, but he did it in dark room as well as in the light also. But he found that this green leaf can restore the fouled air or whatever the organism, the mouse or the burning candle utilized or fouled air, that was restored by the green plant only if sunlight is present. He also did one more experiment. He took an aquatic plant like hydrilla and he kept it in light, and he found that only in presence of light the bubbles were coming out of the green plant. So those bubbles he later identified as oxygen. So though oxygen was discovered by Priestley, Jan Ingenhousz only found that oxygen is released during photosynthesis. Then in 1854, that means almost a century after 80 years, Julius von Sachs, a scientist, he found that the plants prepare glucose after photosynthesis, and this glucose is stored in the form of starch. He also found that the green color pigment, that is now we know it is chlorophyll, he did not know that it was chlorophyll, that green substance which is needed for photosynthesis was stored in special structures inside the cell, that is chloroplast. He identified that. And he also found that only in this green part of the plant glucose can be synthesized. This Julius Wonsack we will learn in mineral nutrition also, because he is the scientist who's discovered our hydroponics, that is soilless culture, where we can give the nutrients to the water and thereby grow the plants. Then we have T.W. Engelmann. T.W. Engelmann, actually he lived up in the beginning till the beginning of 20th century, so experiments are continuing. He did a very interesting experiment, that is he split the white light using a prism. In class 10 physics you learn, when Newton split the white light using a prism, he got 7 component colors of light. What are they? Violet, indigo, blue, green, yellow, orange and red. Then he passed this light into an algae, that is an aquatic algae, that is called a cladophora. And he wanted to detect the oxygen evolution, but we cannot see the oxygen evolution directly, so what he used was aerobic bacteria. You know, aerobic bacteria like to live in the presence of air only, so they will go to the place where oxygen is available. And when he used aerobic bacteria, he found these bacteria were accumulating in the blue region and the red region, because now it's not a white light given. He split the light and he could locate wherever the blue light is striking and red light is striking, there he found more bacteria accumulating. So he understood those parts are releasing more oxygen, which in turn indicates that photosynthesis is more in the presence of blue light and red light. When Engelmann used his experiment, did his experiment using different colors of using prism, then he created the first action spectrum. Action spectrum means the rate of how the photosynthesis is happening, or in which pigment it is higher, that is which light it is higher, whether it is in blue light or red light, that shows the action spectrum. We will learn about action spectrum later. By the middle of 19th century itself, we got an overall idea about photosynthesis, what is happening in plants. They identified, or they understood that plants utilize sunlight, and in the presence of sunlight they make glucose or the carbohydrate using inorganic substances like carbon dioxide and water. Based on this, the empirical formula also they formulated, that is carbon dioxide combines with water in the presence of light to form carbohydrate. Carbohydrate general formula: carbohydrate has carbon, hydrogen and oxygen, and it is actually N times, means whatever carbohydrate you take, they will have the general formula CH2O N times. For example, C and O will be the same number, H will be doubled. I'll give you an example. This is glucose. You know, carbon and oxygen both are 6 only, but hydrogen is double that is 12. So this is the general formula of any carbohydrate. Glucose is the simplest form of carbohydrate, and oxygen is also released during this process. A milestone contribution to all these was given by Cornelius von Neal, a microbiologist. Since he was a microbiologist, he was studying with the bacteria. He used a green and purple sulfur bacteria. They used some source like a hydrogen sulfide instead of water. So basically here what is happening in photosynthesis, carbon dioxide in the presence of water becomes carbohydrate and oxygen. Here you see carbon dioxide has carbon and oxygen, whereas carbohydrate has carbon, hydrogen and oxygen. So from carbon to carbohydrate, hydrogen is being added. So addition of hydrogen we call it as reduction. Right. So we can say that carbon dioxide is getting reduced to carbohydrate. So reduction is happening. But for that reduction, a suitable hydrogen donor should be there. So in this case it is water, but it need not be always water, because these bacteria, green and purple sulfur bacteria, they used a hydrogen sulfide, H2S as a source. So if hydrogen sulfide is the one, after this hydrogen is given, it's not the oxygen released out during that process. It is the sulfur compounds released. Understood? But since here it is water, it is oxygen release. So he found that the source of oxygen released after photosynthesis is from water. Before that it was believed that it is from carbon dioxide, because that was easy to comprehend. Carbon dioxide is a gas, oxygen is coming out. So carbon dioxide is taken, oxygen is given out. So from carbon dioxide, oxygen is going out. We couldn't think of a liquid giving oxygen, right? But now we know that it is not carbon dioxide releasing oxygen, but carbon dioxide is getting reduced to carbohydrate using this hydrogen, and this oxygen is released out. That's why if hydrogen sulfide is the source, we don't get oxygen as a byproduct. So here it's a general formula, H2A plus CO2 in the presence of light, 2A plus CH2O, this is carbohydrate, plus water, but there is no oxygen. Understood. So, these three series of processes, series of contributions by different scientists give us an overall idea about the process of photosynthesis. Thank you for watching my video. If you found this useful, please like and also share it with your friends and hit that subscribe button to get notifications about new videos in this channel. Thank you for watching Biology My Passion.